Hello, everyone, and welcome to today's webinar, X9 Real-Time PCR System, an innovative high-capacity genomic system for superior efficiency. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Standard BioTools. To learn more, visit standardbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Luca Boveri, PhD, Director, Global Product Management, Microfluidic Instruments, and Michael Gonzales, PhD, Director of Systems Integration. Luca and Michael, you now may begin your presentation.
Welcome, and thank you very much for joining us today. At Standard BioTools, our purpose is to accelerate breakthrough in human health by providing the right research tools that can help our customer to generate reliable and repeatable insights. We are excited today to introduce you to our new X9 Real-Time PCR System. The X9 Real-Time PCR System is built to deliver high-capacity genomic analytical capabilities and superior efficiency and efficacy to support laboratories across all the phases of their discovery journey. This innovative and unique platform is empowered by Standard BioTools' proven and proprietary microfluidic technology that enable researchers and scientists to obtain more data and insights with less efforts. Standard BioTools' integrated fluid circuit, or IFC, technology empower life science research by automating PCR reaction in nanoliter volumes to deliver cost-effectiveness, enabling high-throughput productivity with individual reaction simplicity for maximum flexibility and versatility, delivering an integrated workflow for minimal operator contribution. The X9 Real-Time PCR System is the workhorse needed when laboratories demand high productivity. X9 is capable of exceptional processing performances, both in the case when high-volume sample testing is needed, as well as when large target complexity is required. The X9 system, in combination with dynamic array offering, is capable to generate up to 9,216 data points in a single run, more than 46,000 results in a work shift, and simultaneous individual identification of up to 96 targets with no need for complex single-well multiplexing setups. The X9 is a standalone system that does not require any ancillary platform. All the processes that includes priming of the dynamic array IFC, reaction setup, and real-time PCR or endpoint reading are integrated and consolidated in a unique workflow that take place automatically at the press of one single button. The intuitive and user-friendly interface enables immediate implementation in the laboratory routine and workflow with minimal training of the operator that can quickly operate the system with confidence. The fully integrated workflow reduces operator contribution to help maximizing the use of laboratory resources and minimizing human error. X9 system is characterized by standalone capabilities with no need of a computer attached to the platform, and in conjunction with a small footprint enables laboratories to ensure best space utilization. The analysis of the data can be easily conducted on a separate computer everywhere. The powerful Standard BioTools real-time PCR software package supports large data analysis for multiple applications with color-coded expression heat maps, allele maps, and cluster plots for single or multiple runs. The integrated fluidic circuit, or IFC, forms the backbone of the X9 system, supporting different configuration of sample and assays for multiple applications. Each dynamic array IFC is precision manufactured to exact standard of performance and reliability. Dynamic array IFCs empower genomic research by automating PCR reaction setup and real-time PCR processing in nanoliter volumes. These reduce reaction volume, resulting in using less of your precious sample and assay reagents and helping lowering the cost while keep achieving high-quality, consistent results. By adopting different dynamic array formats that are characterized by different samples to target ratio or by integrated additional capabilities, such as a preamplification step, the user can instantly adapt to laboratory immediate experimental needs with no hardware changes required. For example, for high sample throughput and multiplexity, the operator can select the 19224 dynamic array format that can process up to 1,152 samples per 8-hour working shift and have each samples interrogated for up to 24 assay per run. Or, in the case preamplification is needed, for example, when the sample starting quantity is scarce, the user can immediately shift to use 9696 Preamp IFCX that integrates preamplification capabilities automated into the workflow. When superior data generation capacity is required, the scientists can switch to 9696 dynamic array that, by generating more than 46,000 results per shift and 96 assays interrogated per sample per run, supports deeper sample profiling and greater insights into the model that is studied. When a lower data output is required, for example, in the early phase of discovery or low volume testing season, the user can adopt a 4848 dynamic array format that can process up to 48 samples and approximately 2,300 data points per batch. The X9 system, in conjunction with a dynamic array, supports the most popular applications, including genotyping and SNP detection, gene expression and relative quantification, pathogen detection and present substance, and copy number variation. The system is compatible with the most common chemistries on the market and supports the development and creation of custom assays, creating a wide range of testing and investigation options. X9 is compatible with Advanta Sample ID panel. The Sample ID panel supports biobank and genomic centers by providing a high-throughput solution for molecular sample identification, enabling to simultaneously perform molecular fingerprinting, quality assessment, and integrity confirmation on every sample. X9 also supports our Standard BioTools recent release pharmacogenomic panel that allow to process SNPs and CMVs from up to 96 samples in a single workflow with the option of using extraction-free buccal swabs, achieving results in approximately 4 hours with actionable content that can be customized to meet your needs. If you want to learn more about Standard BioTools pharmacogenomic panel, we invite you to join the upcoming dedicated webinar conducted by my colleague Bill Hunt on this same channel on November 30th, or to visit our website. Integrated fluidic circuit technology is the core of Standard BioTools microfluidic solution. This unique and proprietary technology has been proven on the market with hundreds of publications since the release of our first microfluidic platform in 2006. Standard BioTools microfluidic approach empowers you to easily tailor a genomic analysis approach that is well suited to you, to your goal, and objective. Automated reaction setup minimizes operator contribution and reduces human error. Multiplex data output with a singleplex simplicity enables you to instantly add or remove target and assays to immediately respond to experimental needs. Miniaturization of the reaction setup reduces experimental cost of reagents and consumable utilization. In regards of this last point, when compared to conventional real-time PCR microtiter-based approach, X9 and IFC technology can definitely deliver incredible advantage to laboratories looking to minimize cost while still supporting their high throughput and high capacity needs. And this is an example. In order to provide the same data output of a single 9696 format, dynamic array IFC, the user would need 96 individual 96-well microtiter plates. The cost advantage in terms of reagent consumption is evident, as well as the reduced plastic consumption, but I also invite you to think in terms of time to results. With our 9696 format, we are able to obtain 9,216 data points, that is 96 samples interrogated for 96 independent assays in 1 hour and 35 minutes for gene expression application, and below 1 hour and 45 minutes for genotyping. X9 Real-Time PCR System well fit in multiple and diverse research areas and laboratory settings. The high throughput and high capacity capabilities in conjunction with a streamlined workflow supports the need of high volume routine testing in areas like agricultural genomics or biotechnology and fisheries management. High throughut sample identification with Advanta Sample ID in conjunction with integrated reaction setup and X9 system automated operations benefits genomic center, biorepositories, and centralized cell line banks, ensuring accurate sample tracking on quality from acquisition through data reporting. Flexibility of format and configuration in conjunction with the capability to easily switch across application enables research laboratories to easily adapt to changing experimental needs. Now, Michael Gonzales will now take the stage and introduce you to the more technical aspects related to X9 Real-Time PCR System and Standard BioTools microfluidic technology.
Okay, thanks, Luca. At this time, I would like to go into a little bit more detail about microfluidic PCR, the benefits it can provide, and how it is implemented on the X9 system. The Standard BioTools system uses our dynamic array IFCs to automatically assemble individual nanoliter scale PCR reactions. On the dynamic array IFC, reaction assembly starts with the transfer of samples and assays to the nanoliter scale sample and assay chambers shown in the diagram to the right. Each pair of chambers can then be isolated and mixed to assemble an independent singleplex real-time PCR reaction. Up to 96 individual reactions can be assembled per sample, each with a different real-time PCR target. After assembly of reactions, the X9 system automatically proceeds to thermal cycling and data collection. The X9 system with dynamic array IFCs enables automated assembly of up to 9,216 individual nanoliter scale real-time PCR reactions with minimal hands-on time and pipetting, while allowing for maximization of efficiency and cost saving through reduced reagent volumes. Microfluidic real-time PCR offers a number of benefits over methods that use 96 or 384 well plates. One major advantage is the number of samples and targets that can be processed in a single run. With plate-based real-time PCR, the number of targets is typically limited to at most 6 per sample reaction. Any additional targets would require additional reactions with the same sample. Using dynamic array IFCs on the X9 system, up to 96 targets can be assessed per sample with up to 96 samples per run. This means that it would take at least 16 96-well plates, each run with a 6-plex multiplex reaction to generate the same amount of data that can be obtained from a single run on the X9 system. This is a best-case scenario and assumes that all reactions could be run as 6-plex reactions. In reality, this is highly unlikely, as multiplex real-time PCR reactions require extensive optimization that often demands compromises between performance and target throughput. Unlike plate-based approaches, however, dynamic arrays assemble each reaction as a singleplex reaction, eliminating the possibility of adverse assay interactions and the need for cumbersome multiplex optimization studies. Additionally, because each singleplex reaction is isolated, individual targets can be easily added, changed, or substituted without requiring re-optimization of the unchanged targets. Finally, because the X9 system ultimately uses real-time PCR for its detection, it can provide the same high-quality data expected from plate-based studies. The X9 system allows the use of both pre-designed and custom target content. This flexibility allows a wide array of applications to take advantage of the higher assay throughput enabled by the X9 system. Examples of applications on the X9 system in various fields are shown on this slide, and they include, but are not limited to, sample identification and tracking, genotyping-based pharmacogenomics, gene expression profiling, and pathogen detection. On the X9 system, these applications are executed similarly to their plate-based analogs, but they take advantage of the increased sample and target throughput enabled by microfluidic reactions to provide much more data than is practically possible with plate-based approaches. For example, the sample identification panel is available for use on the X9 system that can enable genomic identification and tracking of human samples in biobanks and research studies using a specifically selected panel of 96 SNPs. The ability of the X9 system to assess all 96 SNPs per sample allows confident assessment of sample identity and integrity for up to 96 samples at a time in a single run. To realistically implement a similar screening using a plate-based real-time PCR would require the use of far fewer targets, compromising the ability to confidently identify samples, or dramatically reduce the sample throughput to only a few samples per run. Another panel available, in this case targeting 74 SNPs and CNV loci of genes involved in metabolic processes, permits pharmacogenomic analysis of up to 96 samples at a time with the simplicity and accuracy of real-time PCR-based workflow. Similar to the previous example of the sample identification panel, the X9 system allows the panel to include all targets required to generate a complete picture of the relevant genotypes without compromising on the number of samples that can be processed in a single run. Gene expression profiling is another common application on the X9 system. One example of this is the Standard BioTools immuno-oncology panel, a 170 target panel to assess the expression of genes involved in response to immuno-oncology-based therapeutics. This type of targeted expression profiling can generate deep data sets on a large number of samples that would not be practical with plate-based approaches, all while avoiding the processing and analysis infrastructure required for a sequencing-based method to get the same results. Multipathogen detection is another application where microfluidic approaches can allow deeper analysis than would be practical with plate-based PCR. In this case, instead of running each sample through a series of tests and plates, each directed to a specific pathogen of interest, the X9 system can be used to run each sample against a panel of assays targeting all pathogens of interest in a single run. This permits high sample throughput without the end user having to switch assays from run to run. In addition, the use of single-plex microfluidic reactions for this panel means that individual assays can easily be added, changed, or removed based on changing pathogen and identities without the need to re-optimize the remaining unchanged assays. Well, these are just some of the applications enabled by the high sample and target throughput of a microfluidic PCR in the X9 system. They highlight the diverse array of methods that can be enabled by this technology. The open nature of content on the dynamic arrays means that any application can really take advantage of the benefits of microfluidic PCR on the X9 system. The X9 system allows generation of high-density data with results from up to 9,216 reactions in a single run. This, of course, isn't going to be useful unless there are tools available to easily analyze this large amount of data. Standard BioTools analysis software packages allow for easy analysis of real-time PCR data from the X9 system. This includes automatic assessment of amplification quality and threshold application for CT calling, automatic normalization and delta-delta CT calculations for quantitation and gene expression analysis, and automated SNP and CNV genotype calling for genotyping applications, all to simplify the analysis of large data sets. The analysis software also permits in-depth assessment of results for each sample, displaying amplification curves and associated data for each individual reaction, allowing the assessment of individual reaction performance. If desired, analysis parameters can also be adjusted manually in the same manner as plate-based real-time PCR, giving the user ultimate control over analysis of their data. Finally, result summaries can be exported to multiple formats, allowing for additional analysis. The X9 system uses microfluidic reactions to generate high-density real-time PCR data. This increase in throughput is achieved while maintaining the same high quality and performance that's expected from real-time PCR. The next few slides will demonstrate some of the key performance parameters of microfluidic real-time PCR in the X9 system for both gene expression and genotyping analysis. Accurate discrimination of differences in quantities of starting material is a fundamental requirement for real-time PCR-based applications. To demonstrate this on the X9 system, a 1.5-fold dilution series of human cDNA was run in triplicate on the X9 system using the 19224 dynamic array IFC. The graph shown here shows amplification curves from this run for an assay targeting GAPDH. If you focus on the zoomed in area on the right, you can see that even 1.5-fold differences in starting material can be clearly resolved using microfluidic real-time PCR on the X9 system, demonstrating the ability of the system to accurately detect even small differences in starting material quantities. With up to 9,216 individual reactions on the dynamic arrays, it's important that each reaction chamber performs uniformly across the array. To demonstrate this aspect of performance, a 9696 IFC was prepared using 96 replicates of a single sample and 96 replicates of a single assay and was run on the X9 system. With this setup, each of the 9,216 reaction chambers will contain the same reaction components and will be expected to generate the same CT value during analysis. The plot shown here displays the amplification curves for 96 sample replicates against the single GAPDH assay from this run. A CT standard deviation of 0.06 CTs between the replicates on a mean CT of 12.4 demonstrates excellent uniformity across reactions, indicating the ability to derive accurate results for all reactions on a given IFC. Individual measurements of accuracy and uniformity demonstrate the basic performance of the X9 system, but the true measurements of a real-time PCR system are overall accuracy and reproducibility. To examine these parameters, 2 experiments were conducted on the X9 system. First, an 8.2-fold dilution series of human cDNA was run in triplicate of the X9 system. Results from this are shown in the plot on the left. Analysis of mean CT values versus the log of the concentration shows the expected linear performance with a CT difference of 1 cycle between beta points. An R-squared value of 0.998 and a slope of -3.23 also correspond to typical real-time PCR amplification performance. Taken together, this data demonstrates the accuracy of the X9 system. In the second experiment, 8 human cDNA samples from 8 different tissue types were run against a panel of 96 assays from the Standard BioTools Advanced Immuno-Oncology Gene Expression Panel. This panel contained 91 gene expression targets relevant to immuno-oncology, along with 5 reference targets used to calculate normalized expression. 2 independent 96 IFCs were run, and the gene expression results of each IFC were compared to each other. For the comparison, normalized expression values were calculated using the 5 reference targets and a universal cDNA as a reference sample. Normalized expression values were compared across the 2 runs, and the results are plotted on the left-hand chart. Each point represents the results of a single sample and assay combination on each IFC. In this example, perfectly identical results would result in all points falling on a diagonal line passing through the origin with a slope of 1. When the 2 runs are compared, they generate an R-squared value of 0.997, and all points fall close to a line passing through the origin with an actual slope of 0.98. These results demonstrate highly reproducible results across the 2 runs, and together this data demonstrates that the X9 system is capable of generating the highly accurate and reproducible results expected with real-time PCR. Performance in genotyping applications requires somewhat different parameters than those required for real-time PCR measurements of target abundance or gene expression. However, the underlying requirements of accuracy and reproducibility remain true. To demonstrate the performance of the X9 system in genotyping applications, 96 SNP genotyping assays were run against 92 samples and 4 negative controls on a 9696 IFC. The genotypes for each of these samples for all 96 SNPs were assessed were previously determined from publicly available sequencing and array data. The results of this run show that once accounting for the 4 negative control samples, that of the 8,827 SNP genotypes calls made, 8,826 matched the expected genotype. This means that of all the individual reactions on the dynamic array, 99.95% of the reactions generated a call, and of those calls, 99.99% matched the known genotype of the sample at that loci. Overall, these results demonstrate the ability of the dynamic array in the X9 system to generate large volume of genotyping data with accuracy and an extremely high success rate of individual reactions. Microfluidic devices give the capability to integrate several molecular biology processes onto a single device. In addition to assembling individual qPCR reactions, other automated steps can be incorporated into their design. The 9696 GT Preamp IFC X is one such IFC. This IFC is capable of performing an automated preamplification step prior to qPCR detection to facilitate the use of low concentrations of sample input while maintaining the same ease of use and high throughput of the other Standard BioTools IFCs on the X9 system. The Preamp IFC X allows automated processing of up to 96 samples and 96 targets with inputs as low as 2.5 nanograms per microliter, still generating up to 9,216 genotype calls in a simple 2.5-hour hands-free run. To demonstrate the performance of the GT Preamp IFC X, a run was performed with replicate samples at 2.5 and 250 nanograms per microliter. These input concentrations result in a total of 6.9 and 690 nanograms of DNA loaded onto the IFC, respectively. The SNP genotyping cluster plots show typical performance for one of the 96 assays tested. Tight clustering and clear separation of genotype calls, denoted by the red, green, and blue clusters, demonstrates the accurate results obtainable on the Preamp IFC X. Furthermore, zooming in on one cluster demonstrates a tight overlap of results regardless of starting input, as shown by the circles and diamonds, indicating the individual input concentrations. As demonstrates, the GT Preamp IFC X can produce accurate SNP genotypes across a wide range of input concentrations, demonstrating its utility for low input DNA samples. Overall, microfluidic real-time PCR on the X9 system can be implemented with sample and assay throughput options to meet multiple needs. The open nature of the platform allows flexible content design to meet a wide range of application needs, and microfluidic real-time PCR reactions allow for generation of deep data sets without sacrificing the quality and precision expected from real-time PCR. Thanks for all your attention today. I'll pass it back to Luca for some closing remarks.
Thank you very much, Michael. We have learned today how
can be implemented with sample and assay throughput options to meet multiple needs. The open nature of the platform allows flexible content design to meet a wide range of application needs, and microfluidic real-time PCR reactions allow for generation of deep data sets without sacrificing the quality and precision expected from real-time PCR. Thanks for all your attention today. I'll pass it back to Luca for some closing remarks.
Thank you very much, Michael. We have learned today how the X9 Real-Time PCR System can deliver more data, deeper insights with less effort, exhibiting the blend of features and characteristics that are peculiar to the unique Standard BioTools microfluidic offering, such as nanoliter reaction volume for cost effectiveness, high throughput productivity with individual reaction simplicity, an integrated workflow for minimal operator contribution that reduces human error and minimize operational cost. The X9, in conjunction
Thank
with
you,
the
Luca and Michael, for your
offering, boosts data generation capacity while delivering superior operational efficiency and efficacy. Thank you very much for attending this session today. If you want to learn more about the X9 Real-Time PCR System, please visit our website or reach out to our Standard BioTools representative in your area. Thank you.
Thank you, Luca and Michael, for your informative presentation. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box and click Send. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, Michael, is SNP genotyping an endpoint or real-time PCR readout?
Yes. SNP genotyping on the X9 system can actually be analyzed in both real-time and endpoint modes. Real-time data is collected for all runs on the system, and then the user has the ability to analyze that data in whichever manner they see fit. So in a real-time analysis mode, the user can analyze amplification curves, independently determine optimal cycles for SNP genotyping calls, and utilize all the capabilities of a real-time analysis. Or alternatively, they can use the endpoint analysis as their application needs arise.
Great, thank you. Michael, which qPCR detection, Eisen detection method are compatible with the X9 system?
Most primer and probe-based detection methods are compatible with the X9 system. It uses ROX as a reference dye, and then it supports target detection in FAM and VIC or related fluorophores.
Great, thank you. And it looks like my next question is for Luca. Luca, during the presentation, you mentioned that the system can load different dynamic array without hardware modifications. There will be any collaboration needed, and in general, any periodical collaboration is needed or required by the manufacturer.
Oh, okay. Good question. So dynamic arrays are offered in different formats and configuration to basically adapt to the different laboratory needs or shifting project requirements, right? So all dynamic arrays that are compatible with the X9 system do not need or have the necessity or impose the necessity to modify the hardware components. For example, no need to change the thermal block or the optical system, or make any other adjustment when you move, for example, from a 4848 format to a 9696 or a 19224. So the dynamic arrays are loaded in conjunction with a dedicated interface plate that ensure perfect fitting and functioning of the overall system. Both the interface plate as well as the dynamic array IFCs are barcoded, and the actual X9 system automatically recognizes the format that you are loading on. So in regards of calibration and maintenance, X9 system does not require any specific periodic recalibration, and you can immediately start using the system after the installation, as the platform is factory calibrated already for all compatible dynamic arrays and fluorophores. Now, your laboratory, I would say, operating procedures for various reasons might require your platforms to undergo periodical proactive maintenance, right? So Standard BioTools offers a variety of service plans to best match individual laboratory needs. I would say that if you're interested to learn more, I invite you to visit our website or to reach out to your local representative that can definitely explain you in great details all the options available.
Great, thank you. Luca, you also mentioned the capability to obtain multiplex data with single-plex simplicity. Can you expand on that?
Yep, sure. Setting up a multiplex reaction can be challenging and definitely time-consuming, in particular as the number of targets increase. In conventional multiplex PCR, usually 2 or more primer sets and probes are included into the reaction mix, right? So with this approach, it is possible to quantify and detect multiple targets in the same tube. Multiplex reaction, on the other side, can be complicated to set up. Usually are limited to 2 to 6 color, often requires more expensive dedicated supermix, and are in many instances less sensitive compared to single-plex equivalent. Furthermore, with conventional real-time PCR multiplexing, it is usually not possible to add or switch targets without the need of reassessing and or redeveloping the overall multiplex assay. With IFC technology and X9, you can actually assay up to 96 target per sample in individual single-plex reaction. Each assay can be added or switched or removed based on needs, and there is no interaction across the different amplifications. Furthermore, I would say that another thing to consider is that by using nanoliter reaction volumes, the IFC technology also help reducing experimental cost and save precious sample, for example. I hope I answered the question, but in case more details are needed or you want to learn more about how X9 might be able to specifically support your immediate, I would say, laboratory need, I encourage you to reach out to our extremely competent and knowledgeable sales team that I'm sure that would be able to help you with more details and specific for your specific cases.
Great, thank you. It looks like we have one more question here. How do the microfluidics work on these plates? How do you pipette in the samples and the probes?
Yeah, so our microfluidic devices are embedded into a plastic carrier that's the same footprint as a 384-well plate with the same well spacing as a 384-well plate. So the samples and probes are individually pipetted into those inlets on the carrier, on the IFC, typically using a multi-channel pipette, which 8-channel pipettes make the pipetting very easy. And that's really it. Then you put that microfluidic device into the system, and the X9 system itself handles all of the microfluidic workflow on the plate. It pushes the samples and assays in and does all the microfluidic processes automatically.
Great, thank you so much. It looks like that brings us to the end of our Q&A portion of the webinar. Michael and Luca, do you have any final comments for our audience today?
No, Megan, I don't have any final comment. I just encourage all the audience or the people that attended this webinar, if there's any additional question, again, as I know that already said that, please reach out to the sales representative of your area, and we'll be happy to provide you with all the details and more information or specific information based on your specific laboratory needs. So thank you again for joining us today, and have a good continuation of your day.
Yes, thank you again, Luca and Michael, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Standard BioTools, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us and for their interesting questions. Questions we did not have time for today, and those submitting during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues that may have missed today's live event. Until next time, goodbye.